Good afternoon, everyone, and welcome to Research Conference. Today, we are very excited to have Dr. Kimberly Blackwell joining us. She is Professor of Medicine and Assistant Professor of Radiation Oncology here at Duke. She received her undergraduate degree in bioethics here and completed her medical training at Mayo, had enough of the Minnesota weather, and came back for internal medicine residency here and a hematology oncology fellowship before joining the faculty in 2000. Dr. Blackwell is one of the nation's leading breast cancer researchers and has played a major role in developing therapies for revolutionary non-chemotherapy-based approaches for treating breast cancer. Her work on promising new therapies that selectively target breast tumor cells led to her inclusion on Time Magazine's 2013 list of the 100 most influential people in the world. More recently, her work has led to the approval of the first biosimilar for treatment for cancer in the United States, and she's served as the principal investigator or co-PI on more than 50 cancer clinical trials. We're very excited to have her today, so please join me in welcoming Dr. Blackwell.
Well, thank you, Emily, for having me. Can everyone hear me okay? I'm usually really loud, but I very rarely come all the way over here to North Pavilion. I didn't know what was lurking in the basement of the building, so I didn't know exactly what was going to come of the audience today. But what I am here to discuss is really what I think is one of the potential paths forward for innovative and hopefully expedited cancer drug development. As most of you know, cancer is a global problem. Many research funders like BCRF and Komen in particular are looking for global avenues to develop not only cancer epidemiology, but novel drug development strategies. And for those of you who have done therapeutic trials for a while, with the continued complex regulatory environment and patients that find it perhaps difficult to get access to clinical trials, even with the Moonshot Initiative and Dr. Biden or Joe Biden out saying we need to get people onto clinical trials. At least in my now 10 years or 20 years of doing drug development, we've really struggled with finding strategies to accelerate getting new drugs to patients. And I always start a talk with saying that, my primary focus is figuring out how to give patients therapies that they couldn't get anywhere else, at least here at Duke. And for those of you who have had either loved ones or friends or family, or I guess family members would be loved ones usually. That was my joke for the day. I hope you caught it. I'm not a joking soul, but I thought that was kind of funny. Those are not mutually exclusive. Usually they overlap. You realize that it's not due to the patient's inability or desire or lack of desire to help us make new treatments. The thing that I'm impressed with every day is how generous my patients are with their time and the whacked out stuff, for the lack of a better descriptor, that we ask them to do. And I ask myself frequently every day, if someone asked me to do this and my time on the planet was limited, would I actually sign up for it? And I think that really should be a foundation for everything we're doing, which is it high quality? And I'm just talking about the cancer space. Would you be willing to do it? And does it offer enough promise? But more importantly, does it have a strategy to getting the drug approved? Because I think we do a lot of trials that don't have an executional strategy for big pharma, little pharma, or even the poor scientist who's trying to start their own drug company. So that's my 2 cents worth, which is the patients want to help us. I perceive, this is my other political statement, that in a transparent environment, big pharma wants to help us too. And it's a matter of finding common goals that will serve both the industry effort as well as the scientific effort. And so what I'm going to describe over the next, hopefully 35, 40 minutes, leaving some time for questions, is really what I'm pretty excited about right now at this point in my career, which is this idea of how do we accelerate both the science and drug development, in particular in a pure regulatory sense, in a way that's different than the way we've been doing it. Because something's got to change in cancer. I don't know any other field but breast cancer. But I have to say that the median life expectancy for metastatic breast cancer has remained at 24 to 27 months for the past 2 decades. So for those of us who, walk the Coleman race and buy the toilet paper with the pink ribbons on it and buy the mascara that, goes to fund BCRF, we as constituents also should be asking the question, what are we doing that's not working and how can we change that? And this is just a small, I'm always about the low-lying fruit. This is a very small effort to try to do things a little differently. And I think we're in a good, if not an excellent position to do things differently, at least in this space. So again, over the next 35 minutes, I do want to give you, some people who are involved in the trial are actually in the audience, so I do want to give you some kind of broad overview of the field of triple-negative breast cancer, where we're at, and some of the more promising treatments available for triple-negative breast cancer. But more importantly, I want to give you a kind of a foundation for how we're developing drugs within this space. I'd say they're large, they're not necessarily inherently based in science or predictive biomarkers. And then I'll finish the second half of the talk to really talk about the history of this Singapore collaboration I've been working on now for close to 2 years, the trial that's embedded in the DCRI that's about to start, hopefully, and really where I think this maybe meets some of the goals that I'm trying to achieve as a therapeutics clinical trialist. Okay, so I made an assumption that none of y'all knew anything about breast cancer, so I apologize if you're the world's expert in breast cancer, but this is really where we all start, which is, when I started, breast cancer was breast cancer, and everyone got some chemo, and everyone got some anti-estrogen therapy. And at least in 2000, about half of the patients weren't even having their estrogen receptor tested. Now, we did it here, but if you were in Fayetteville or Lumberton 16 years ago, we just said you had breast cancer, and you got chemo, and then you got an anti-estrogen pill, which seems kind of absurd now because we knew that there were probably subtypes of breast cancer. We definitely know it now. And just for the premise of this talk, I'm really going to focus on this subset, this subset of breast cancers that we affectionately, this subset of breast cancers that we refer to as TNBC or triple negative breast cancer. And I'll show you the numbers here on the next slide, but about 80% of all breast cancers either are dependent on the female hormone estrogen, we term that estrogen receptor positive, or they're addicted to an oncogenic signaler or surface receptor known as HER2. And for those of you who think a lot about the history of breast cancer, which I do, but very few people do, the first targeted therapy in any cancer was described in 1896, and it was actually George Beecham who was removing the ovaries of sheep in northern Scotland and found out that it affected lactation. And he went back, ran down to London and took they weren't even called ovaries back then, these organs out of females who clearly had locally progressive breast cancer. And lo and behold, 2 out of the 3 women had responses. That's actually the first described in the literature targeted therapy for any cancer. So we have that. I'm kind of proud to be a breast cancer expert, in case you haven't figured that out. And then one of the top 10 medical discoveries in the 20th century was the invention and ultimate approval of a monoclonal antibody against the HER2 protein known as trastuzumab or Herceptin. And that's a very effective strategy for that 1 out of 5 women facing HER2-positive breast cancer. So just to try to lay the landscape of breast cancer, we now have and I'm going to talk about the subtypes of triple negative here in a second, but for breast cancer, kind of 10,000-foot view, we have 3 types of breast cancer. And in 80% of the breast cancer, we have Herceptin, trastuzumab, and anti-estrogen strategies. We now have CDK inhibitors, which have been shown to be highly effective in ER-positive breast cancer. So in 80% of all breast cancer, we have therapies that have been proven to improve overall survival and have been proven to significantly reduce a woman's chance of her cancer coming back. This one is the one I'm going to focus the rest of the time on. This is triple negative, and to date, I'll say this about 3 times throughout the talk, we have no effective strategies for triple negative breast cancer. I'll show you some pretty sobering figures here in a minute. And this has been recognized by the FDA for the context of this talk as an unmet need in the cancer space. And it has led to 3 breakthrough status designations for drugs within the space, and I'll speak to those here in a second. So let's see here. So, when you do a pie graph and you're left with 20% of all breast cancer, you think, how big of numbers is that? It actually is about the same amount of patients each year, well, 9,000 less, of people who are facing pancreatic cancer. It's, about 50% the amount of patients facing primary brain tumors. It's a larger number of patients than patients facing ovarian cancer. So from a pure pharma and drug development standpoint, this represents a fairly sizable number of patients that are affected by this disease each year. And I just wanted to put that in perspective. The things we know about triple-negative breast cancer is the median age of diagnosis is 12 years younger than the 80% of all the other types of breast cancer. And we're not quite certain what that is, why that is. I published on it, and I'd be the first to say that we don't know. It also differentially affects African Americans, especially of Northern African descent. And then, as many of you know, because I just got reminded me of Angelina Jolie's getting a divorce from Brad Pitt, in case you didn't hear that about an hour ago. This gene mutation, yeah, I know, it shocked me too. And I see the audience is like, oh my goodness, why didn't I get that notification? She carries a gene known as BRCA1. It's a mutation that you're born with. And basically, if you have this mutation, the BRCA protein cannot repair your DNA and put you at especially high risk for breast, prostate, and ovarian cancer. And this is the type of breast cancer that's very similar to ovarian cancer, and is, if someone carries a BRCA1 mutation and develops breast cancer, for all practical purposes, they get triple-negative breast cancer. So it's kind of like the double whammy. Not only are you born with an unfortunate deleterious mutation, but then you get the worst form of breast cancer that we don't really have any good treatments for. And that's why we're so aggressive with preventative and prophylactic measures in BRCA1 in particular. There's another gene called BRCA2 that's associated also with hormone-sensitive breast cancer, but BRCA1 is the one where you get triple-negative breast cancer and ovarian cancer. And the things about triple-negative breast cancer is that the risk for recurrence across the board, independent of stage, is about twice that of the other forms of breast cancer, especially with the introduction of effective therapies. And relevant to trial design, relapses, 90% of relapses recur within the first 2 years. So again, just from a pure trial standpoint, trials that are looking at what we call early stage triple negative or adjuvant therapy read out very quickly, because unfortunately the relapse rate is quite high in this population of patients. It's kind of, if you think about pancreatic or lung that goes to resection, and we don't really have very good, I would say, effective strategies for preventing those cancers from coming back after a complete resection. Triple negative represents a similar type of recurrence. It also, my other interest is really developing novel therapies for the treatment of brain mets. 60% of all people facing metastatic cancer will die with brain mets, and the only therapy we have is radiation. So that's a whole nother area that needs to be explored, and unfortunately brain metastases are quite common in triple-negative breast cancer. So what are the standard treatment options currently for metastatic? Oh, and so for the rest of the talk, I'm going to talk about metastatic triple-negative breast cancer. That's otherwise known as incurable. It means that the goal is palliation. I hate that word. I probably shouldn't say that out loud, but it just has so many connotations. It means we don't have treatments. But the reality is the goal in treating metastatic triple-negative breast cancer right now is not for any type of curative intent. It is hopefully to prolong the highest quality of life for as long as possible for these women facing this disease. And I kind of put this up there because there's lots of things we use. For the most part, there is not a drug approved at any regulatory agency specifically for triple-negative breast cancer. We've kind of lumped breast cancer together in developing clinical trials. And there is no standard option. So on boards, if you're asked, what is the first-line treatment for triple-negative metastatic breast cancer, the answer could be any of these. And that's unlike the HER2-positive breast cancer space or the ER-positive breast cancer space. So what do we have currently available? I just want to give you two examples of what and how these trials have been going. So this is a trial of all comers, not just triple-negative breast cancer. This was our most recent, I'll make certain I'm telling you all the truth, our most recent chemotherapy drug approval in metastatic breast cancer. It's a drug known as eribulin. It basically is a microtubule stabilizer, so it functions very similar to vincristine, vinblastine, paclitaxel, you name it, those classic drugs that we use in breast cancer. And we compared it to the 5-FU analog, capecitabine. That's an oral chemotherapy. And basically, after 1,100 patients, 3 years, and you guys probably know the cost of a trial like this for the drug maker, we found out that there was no difference between the two drugs. So that's kind of a disappointing equivalence study. We did find out, though, that in this triple-negative subgroup, that the newer drug was better than the oral form of 5-FU known as capecitabine. And this was presented in 2014, and then there was just kind of a whole, and this is one of the beginnings of the trial I'm going to describe to you. There was this whole kind of recognition, I think, both on pharma's part and investigators' part, that for slower growing cancers, it's much harder to see a signal when you're using cytotoxics, except in the triple negative space, so that if you do have a superior agent, perhaps it's more evident in the first line. This is all first line, meaning patients had never been treated for their metastatic breast cancer. Perhaps by studying it only in triple negative breast cancer, you could perhaps see a signal, and that was in combination with the FDA declaring that triple negative is an unmet need. And then all of a sudden, everyone wanted to study triple-negative breast cancer. Drug companies and breast cancer doctors. And I can say that the entire immunotherapy field for breast cancer is being studied in triple-negative because of the FDA designation. And because of this, if you're going to see a signal of activity, you're more likely to see it in triple-negative than some of these subtypes of breast cancer that people live for years and years and years. The HER2 space is completely crowded. It's almost an impossible strategy or landscape to think about a regulatory strategy for drugs right now, because the median survival for HER2 positive metastatic breast cancer is actually 59 months. That's great. I mean, I love that. That's the impact of these, HER2 targeting agents. But imagine trying to develop a drug within that space when the median overall survival is close to 5 years and the FDA requires a survival benefit. I mean, those are kind of like cardiovascular trials, and we don't have that. I guess we don't have that patience as cancer doctors. So this was one of the first, I'm only going to show you 2 studies in this space, but it got us all thinking about, if you're going to look for an effect, do you do it in every form of breast cancer, which is how we've been doing it for 40 years, or do you subset these subsets of breast cancer in hopes that you might be able to tease out and have a higher level of sensitivity for drug development? And this is the only other trial I just wanted to show you. And again, this is a trial specifically in triple-negative breast cancer called TNT, which I think is one of the best cancer drug trial names I've ever seen. This was all triple negative and ER positive if you had this BRCA mutation. And it was kind of an interesting study. It was powered on overall response rate, so automatically it's not a drug company sponsored study, it's a foundation sponsored study conducted in the UK. And they basically did carboplatin for half the women first and docetaxel, which is a taxane. Platinum is a DNA damaging agent used in many types of solid cancers. Never approved at the FDA, been off patent forever, like forever. And then they switched to docetaxel or carboplatin. Here are the results of the study. And this kind of sets some of the background for the study I'm going to go over with you. This has actually just been published in Lancet. But once again, another equivalent study, this time in a specific population of first-line triple-negative breast cancer. That one chemo was no better than the other chemo in triple-negative, so a little different than the first study I showed you. But there was this interesting subgroup, and these are women that are women whose tumors are inherently sensitive to DNA damaging agents, because I've already alluded to that the BRCA protein is dysfunctional, they can't repair their own DNA. So radiation works great if you can give it to wherever the cancer is. I wish we could give it head to toe. That would solve a lot of problems. And you could see that the response rate to platinum in these BRCA mutation carriers was double that of the docetaxel. And this got us thinking a lot about DNA damaging agents in the setting of BRCA mutations in particular. So that is what we have right now. So when I showed you that list and said you could pick any of them, you really can pick any of them. And that also has made drug discovery and development in this space a little challenging, because if you don't have a gold standard, and this has actually been an issue for our trial, even though it's a smaller trial, if you don't have a gold standard, what do you base your go, no-go decisions on, right? If you don't have a historical standard for PFS or overall survival first line, because everyone gets different stuff, it's very hard to convince many agencies, and probably more importantly, drug companies, that you have a good comparator arm, because there's a lot of uncertainty about the comparator arm within the regulatory agencies. So what are the emerging strategies? Well, this is where the science hits the road, maybe. I don't know, the rubber hits the road, the science hits the road. And this was a really pivotal paper from Vanderbilt in 2011 for those of you who are interested in the science of triple-negative breast cancer. And what they basically did is they did an RNA transcriptional analysis, very similar to what we had done to breast cancer just in general. And they just looked at triple negatives. So in 2005, Chuck Peru, working at Stanford, subclassified off of a transcriptional analysis of breast cancer into 5 subtypes: luminal A, luminal B, HER2 enriched, not specified in basal, which is pretty much associated with triple negative breast cancer. And then the group at Vanderbilt took a group of just triple negative. So we're kind of narrowing our scope down, and was able to identify 6 subgroups of triple negative breast cancer that they thought were both biologically relevant but offered potential subgroups of triple negative to match up therapeutic options to. And so this is all triple negative, so this is where it gets really complicated, right? So now we have 6 groups of triple negative breast cancer based on a transcriptional analysis. It used to be basal basically meant triple negative, and so if you go to conferences, for those of you who attend cancer conferences, and people go basal, 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 they're really not using the language precisely. It really is now kind of subpar down into basal 1, which is really consists almost wholly of BRCA mutated tumors or other DNA repair genes that are mutated. And I took all the slides out for the sake of time, but we now have 16 other DNA repair proteins that, when mutated, increase the risk for breast cancer, as well as increased sensitivity to DNA damaging agents, at least preclinically. Then there's this basal-like 2, which I'm particularly interested in because we have a very large EGFR therapeutics portfolio, especially within the brain tumor center. And there is a subpopulation of triple-negative breast cancer that's highly dependent on EGFR. What happened is, just to diverge for a second, we studied triple-negative and we put all these EGFR inhibitors on it, small molecule inhibitors and antibodies, and guess what? It didn't work that great, because whatever 5/6 is, 5/6 of the triple-negative breast cancers were not dependent on that particular growth factor. So we're now revisiting that in a number of efforts, but mainly at Duke as a site, to see if we can go back and identify these basal-like 2 subgroup of triple-negative and match them up with some of our novel HER1 or EGFR inhibitors. There was an immune-enriched population of triple-negative, and this is a very interesting triple-negative subset, because they actually do better. And we're not clear, but the simplest explanation is that if you have an immune response to your triple-negative breast cancer, you do better. And it's very interesting, if you were a drug company, would you study your immunotherapy in the tumors that don't have a heavy immune infiltrate, i.e. there's not a good immune response, or would you actually study your immune therapy in a group of triple-negative where you already know there's an immune response going on? And some of the work that we're going to be doing within our context of our trial will hopefully, in a small way, address that. But there are many other efforts going on to see the role of immunotherapy in all these subgroups, but in particular this immune subgroup. And then there's two other groups called mesenchymal, and I'm going to skip over those because I don't really understand it, and there's not a specific clinical trial. I understand it. That was my second joke of the day. But really, it's about an hour-long lecture, and the reality is that there really aren't any trials focusing on these two subgroups. It's very, we have some Notch inhibitor studies in particular that are probably the furthest along in triple-negative breast cancer. Some Wnt in particular, that might be targeting these mesenchymal subgroups. And then finally, I'm just going to show you very quickly the data around this. This is a whole nother field. There's about 7 companies studying androgen receptor antagonists. So if you study prostate cancer, you're going to think we've been copying you in the breast cancer world. But there is some good data around the fact that in subgroups of triple-negative breast cancers, androgen receptor is important. So this is kind of the same slide without the fancy heat plot, and this basically just shows these 6 subgroups and potential ways of targeting with, they're not in the exact same order, but the idea that you could, now we've gone in 16 years from everything's breast cancer to ER positive, HER2 positive, triple negative, to then what Chuck Peru and others have done, which is 5 subgroups of the total breast cancer pie, luminal A, luminal B, basal, HER2 enriched, to now 6 subgroups of triple negative breast cancer. All right. So what does the field look like right now as far as clinical trial options for, again, metastatic triple-negative breast cancer? I'm just going to touch on the first 2 very quickly because they're not involved in our study. But most of you are aware that we've had some clinical successes with this concept in breast cancer. I don't think they're being developed in any other disease states. Known as an antibody drug conjugate. There's all kinds of catchy names for these drugs, the magic bullet, the Trojan horse, I don't know, I've heard about 6 of them. And the work that was done here at Duke by myself and Neil Spector actually led to the first approved ADC for solid tumors known as TDM1. That's trastuzumab, linker, and derivative of maytansine-1. The idea is that you take about 4.5 really toxic stuff, if I can get my pointer to work. In this case, in this drug, it is basically another form of not cathepsin D, sorry, a tubulin inhibitor called MMAE. You don't really need to know what it is. All the things we're attaching these antibodies are really toxic, including radiotherapy. I actually think the radiotherapy approach is going to be as effective as the chemotherapy approaches. But in this case, it's an antibody drug conjugate against this protein called GPNMB. And so we're actually doing a study with Celldex that I am one of the global PIs on, that basically is looking at this antibody drug conjugate only in triple-negative breast cancer, only in the basal subtype that has this high levels of GPNMB right there. And so the idea is this drug had some activity, and I left this slide in for 2 reasons. One, to show you that the antibody drug conjugate's probably worth studying. So people that had high amounts of this protein in triple-negative breast cancer basically had a 3-month progression-free survival, and this was fairly heavily pretreated. I left it in here because it tells you how horribly these patients do. I mean, median overall survival in this trial, which was just presented a 1.5 years ago, is 10 months for patients that basically, and then this is compared to just a chemotherapy regimen. But the bottom line is you can see that these patients just don't do very well, and so we need to do better. And here's the trial that we're participating in right now, which is the ADC versus capecitabine. And unlike that and this is why I left this study in, is because unlike the 1,100 patient study of eribulin versus capecitabine that took us 5 years to accrue to and showed no difference in one chemotherapy versus the other in an unselected population of patients, in triple-negative breast cancer, because of the rapid, unfortunate time to progression-free survival and because of the short overall survival, to get a regulatory or registration strategy in this space, you only need 300 patients. And so this is, I don't know how to say it, I want to get new drugs for triple-negative breast cancer, but if you're a clinical trialist, this is a space that doesn't have approved agents, and you can really get a signal very quickly because, unfortunately, of the poor prognosis of these patients. So this is one study that really is one of the hallmark or priority studies ongoing in triple-negative breast cancer. And then some of you probably have heard about this study. It's an immunomimetics drug. It's an antibody drug conjugate against TROP2, which is a protein that's overexpressed on triple-negative breast cancer. It just got awarded breakthrough status probably 2 months ago, which is a big deal in the cancer arena. And it's because of this. These patients on average had lived through and were hardy enough to go on another clinical trial after 4 different regimens for their triple-negative breast cancer. And to date, this objective response rate of 1/3 or a clinical benefit rate of 2/3 is actually, unfortunately, the highest response rate we've ever seen in a phase 2 triple-negative breast cancer study. So that's kind of the lay of the land as far as clinical trials. The final trial is this idea that we have a subgroup of triple-negative breast cancer, remember the bottom of the heat plot, of triple-negative that's driven by androgen receptor. And traditionally, both in prostate and breast cancer, we haven't had very good antibodies against staining androgen receptor, and we haven't had a good idea of cutoffs. But we know that as high as 1/3 of patients will have overexpression of androgen receptor who are facing triple-negative breast cancer, and as high as 75% of ER positive, that hormone subgroup, will have overexpression of androgen receptor. And for those of you who work in the cancer space, you know there's been large amounts of successes with androgen receptor antagonists and androgen depleting drugs over the past 10 years in prostate cancer, and we're doing the same thing in triple-negative breast cancer. This is the results of the enzalutamide study that was just presented. And basically, it doesn't take a lot of androgen receptor, at least in this study, to qualify. About 89 triple-negative patients were put on this study, and about 75 were evaluable at the time of the most recent analysis. And what you see, again, if you're black or white, like in terms of results person, you might think this is really a shade of gray. But in the intent to treat population of triple-negative breast cancer, we saw 1 out of 4 patients responding to this fairly benign therapy. And so this has renewed a lot of interest in targeting the androgen receptor in breast cancer. So we have 2 trials of androgen receptor antagonist at Duke right now looking at triple-negative breast cancer. And I'll tell you, this has really changed our thinking a little bit, because we were a little bit of kind of heavy hitters when it came to taking care of triple-negative breast cancer. Because these patients progress so quickly, everyone was a little afraid of doing anything but the highest amounts of chemotherapy possible. We had to hit it on the head. We had to get under control, all these kind of war analogies that we're haunted by in cancer. And the reality is that these kind of trials have made us realize that we need to be smarter, not necessarily back to the chemotherapy cowboys we were 20 years ago. So that's really my first 25 minutes on triple-negative breast cancer. I hope that that has demonstrated to you that about 1/5 to 1/4 of all breast cancer is triple-negative. It really does depend on where in the world you look, and at least in China, it could be as high as 1 out of 3 women are facing triple-negative breast cancer. It has a poor prognosis, and even in the first-line setting, I showed you a single study, but this is evident in other studies, the median progression-free survival is only about 4 months. That's pretty horrific when you think about it. And that from the time of diagnosis to the time of death on average is about 14 months in the metastatic setting. There are no approved therapies specifically for triple-negative breast cancer, which makes it an unmet need compared to other forms of breast cancer. All right, so that's the background to the DORA study, which is being supported by the DCRI. And this, I'm going to just tell you like a minute of the history. And I actually think that the fact that Duke has a med school and a presence in Singapore, located in Central Asia, really represents an opportunity for us to collaborate with that part of the world. And so I started fairly small. And the story, the trial is known as the DORA study, and it's durvalumab, which is an anti-PD-L1 immunotherapy. It's an antibody. and olaparib, which is a PARP inhibitor. So I haven't talked a lot about immune therapies. That's a whole nother talk. I have one slide. I'll briefly go over that with you. And the PARP inhibitors basically work by knocking out the PARP protein, which in BRCA carriers, the way I explain it to my patients is if you have a chair that has 4 legs and you're already missing one leg, the chair's probably still standing. You don't die if you have a BRCA mutation. Your cells can repair themselves, but it's a little wobbly when it comes to repairing the DNA. PARP is another leg. PARP proteins also are responsible for DNA repair. So if you inhibit that, the chair falls over. That's exactly how I explain it to patients. And that's kind of, in the simplest of terms, important because what this trial is attempting to do, and I'll get to the trial design in a second, is really capitalize on 2 things. That there's a subset of triple-negative breast cancer that is found in non-BRCA mutation carriers but is inherently unable to repair its own DNA, maybe because of another gene mutation, maybe of something that's yet to be discovered. And we're going to use carboplatin, I'll get to that in a second, to try to select that type of triple-negative breast cancer out. And then the immune therapy, because everyone's doing immune therapy, is, I'll give you the rationale for that for a second. So let me just give you the background to the collaboration and the trial design. So I'm fairly outspoken about how slow things are. Trish can attest to that from our teleconference this morning. But unlike cardiovascular trials, where there were no cooperative groups 20, 30 years ago, and so everyone had to work with the DCRI and everyone had to work with the drug companies, cancer has been a little different because we've had different structures in which to conduct cancer trials. Right now, in 2016, there's really 2 main ways. If you're an investigator and you want to design a trial and you don't work for a drug company, that you actually will go about designing a clinical trial, phase 1, phase 2, doesn't matter what phase. So basically, you can go to pharma who have amazing drugs and amazing options and amazing scientists and basically say, I want to do this. And you go through a very, as you know, a very time-intense process. I'm Kim Blackwell. It's that movie Shark Tale, if anyone has kids and the sharks are getting ready to eat the shrimp on the cocktail and the shrimp are like, please don't eat us. That's Kim Blackwell, and then that's the big drug company, and they might let you free, and they might even actually I don't know what else you do if you're shrimp, but it kind of feels like that at times. And the process is slow. And even when the drug company comes to you with an idea, the process is slow. Even if it's a priority study, the process is slow. The trials tend to be large because, as we all know, the indication for a 3,000 patient study better be indicated for as many cancer patients as possible. So subsetting 1/6 of a 31,000 of a disease that only affects 31,000 patients a year means that market for that particular indication, even if the trial is successful, is only 5,000 patients in the US each year. In general, the trend right now is to skip the phase 2 portion almost completely. And I can give you about 20 examples, especially in the immunotherapy world, because these drugs are being developed in lung cancer and bladder cancer and thyroid cancer. So if you have safety from your bladder registration study, then you just put it in breast cancer and let's do a phase 3. And that's what's happening, is that these are particularly high risk, but very high gain studies, but it takes a long time to accrue to them because of the size of the studies. And then when you're doing that type of study, unlike cardiovascular disease where it's easy enough to get a serum or plasma sample, tumor samples are not collected for a number of reasons. And so they tend to skip the phase 2 portion completely, which is where some of the discovery science really usually happens. I sound like an investigator, but the reality is there's very little investigator input in some of the large registration studies. They listen, they have thought leaders, but at the end of the day, the decisions are made more on the things we've just discussed. And they really lack anything that's a hassle. I don't know how else to say it, whether it be, you have the patient come back to get another biopsy. And I started this talk by saying patients are not the rate limiting step here. I mean, yesterday, I saw 4 new patients. All 4 of them were like, where do I sign up for the biobank? And these people are getting the liver biopsies and their lung biopsies. And for women who have faced breast cancer, a breast biopsy is a real hassle. They don't put you out, they numb you up barely, and you have your breast in some contraption. My patients, when they go for bronch or liver biopsy, they're like, that's the best thing that I've ever done, right? I mean, they knock you out, you don't remember it, it's done in a safe environment. But we don't ask patients to do that in part because of the larger phase 3 nature of the studies. And that's not a criticism, these are all observations. Investigator-initiated is where you really become like the shrimp. I don't even know if you're the ice in the shrimp cocktail. And you have a great idea and you want to have a good scientific question embedded in an investigator-initiated studies. And unlike cardiovascular disease and probably GI disease, we've had these very large cooperative groups where we can do investigator-initiated studies supported by the National Cancer Institute for the most part, some foundation funding. But the problem with these studies is they're even slower than the industry-sponsored studies because they're so poorly funded. And at least at Duke, we constantly are having to ask the question, what is going to be our loss by opening up an NCI-sponsored cooperative group study? We function at a loss across the board for NCI-funded studies. Investigator-initiated studies, and this will probably make the financial people, or Andrew in the front, fall off their chair. Drug companies have this standard, we'll give you $8,000 per patient. You can do whatever you want. Well, we can't even pay an IRB fee with $8,000. That's not the truth, but you see what I'm saying? And so then you're constantly trying to figure out how to do studies that are underfunded. So the premise behind the DORA study is, what if you merged an investigator-initiated study, but funded it at a level that you can embed an ARO, an academic research organization. And again, I think this has been a successful model in other diseases. We've just not been as successful in the cancer space as I think we could have been and should be. And so my collaborators in Singapore and we wanted to do an investigator-initiated study, but the complexities of how do you convince a drug company that this isn't going to be an $8,000 per patient study. We want to collect the same quality of data you would do if you were running this study on your own, but we want to control the study. And for those of you who work over here, my perception, although it might be delusional, is that that is the model that has served cardiovascular space extremely well, and as part of the DCRI model and other disease entities. So the idea behind the DORA trial was really to create an environment that would be sustainable for future trials of a supercharged investigator-initiated cancer trial. And this is my collaborator. This is literally our first meeting at ASCO in 2015 when we came up with this cockamamie idea, which is, she's a Duke faculty member, and all of the MDs that work in Singapore General Hospital and at the National Cancer Center of Singapore have the ability to have a Duke appointment, which has a lot of weight because she has the same rights that I do as a Duke faculty member. She just sees patients in Singapore. And this has really created some opportunities for expedited things happening, because when drug companies see us, they see us as Duke faculty members. They don't see, oh, okay, so we're going to have an investigator here and an investigator here. No, we actually are functioning under a central Duke umbrella, which in principle, and I think actually in reality, should have some benefits for these types of collaboration. We have, for those of you who haven't been to Singapore, we have a med school there. I mean, it looks no different than the med school that we walk into here. And so this really was this idea that we should capitalize on having a Duke-based med school in Singapore in the cancer space. And this had really not been done before. I know there's been some opportunities in vaccine research and perhaps cardiovascular, but we were really trying to build a collaboration in the cancer space and something we knew, which was triple-negative breast cancer. And this is a coffee cup down here, so we both like coffee and shopping, so that worked out really well too. For those of you who don't know much about Singapore, and it looks like the spacing's a little off, it's almost identical structure to the Duke enterprise. So the academic appointments are in the med school. So Rebecca has a faculty appointment within the Duke National University of Singapore, the med school. Sing Health is basically aligned with Singapore General, where the majority of the patients are seen. And then they have a cancer center very similar to the Duke Cancer Institute, where some of the research, sorry, I realize there's spacing on here, that some of the research is homed within the National Cancer Center of Singapore. So in terms of wrapping my head around what this would look like, it's almost identical to the structure we have here. We all have academic appointments. Our research endeavors are within the Cancer Institute. And then there's a large health center enterprise that actually is, seeing the patients and touching the patients. And the other thing that really impressed me about Singapore in the cancer space is that because of the centralized, if you get your cancer care in Singapore, you get it at one of 3 hospitals. It's not like the United States where you get it in one of 3,000 hospitals. So if you are enrolled in a phase 1 study there, the default is we're going to biopsy your metastatic tumor. It's not like maybe, should we, does this trial require a biopsy, does this not? So they're generating this whole population of, I hate to say it, but kind of recyclable patients, because they biopsy the tumor at the beginning. They have a number of precision medicine panels, and they're actually selecting the enrollment into their phase 1 studies based on the precision medicine analysis, which then they put the patient on the trial, but then they have all the other molecular alterations for when that patient unfortunately progresses in the phase 1 study. And that's pretty similar to the way we do it here at Duke. Although if you have breast cancer, you come to the breast cancer clinic. If you have lung cancer, you come to the lung cancer clinic. There, if you live in Singapore and you have metastatic cancer, you are seen in their phase 1 unit, you get your biopsy. It doesn't really matter what type of cancer you have. And if we're going to make progress in precision medicine, many of us believe this is the way to go. And they have this up and running in Singapore, which makes it kind of a great opportunity for us to learn from them, but also to capitalize on the assays that they've already developed within their science space at the med school. Now, this is an old list. I was unfortunately unable to update this, but in 2013, they had 20 molecular-driven trials, which is quite a large number of trials for a single institution, and they're the preferred clinical trial center in Asia for 4 major pharma. So I heard that, in particular, they have strengths in lung cancer, head and neck, and hepatocellular because of the incidence, as well as gastric. Those are their kind of heavy hitters. And because Rebecca has expertise in breast cancer, I think that this collaboration really offers us an opportunity to think about growing what has been so successful in other tumor types into the breast cancer space. So I'm running short on time, so I'm going to go over some of the science behind this trial. I have about 10 or 12 more slides very quickly. I've kind of given you a background on where triple negative space is. This is another genetic analysis of breast cancer that was published about 4 years ago in Nature. And probably the most important thing you need to know, and this was the background for the study as well as some of the other stuff that I've shown you. Here are the PAM50 defined types of breast cancer. So this is typically ER positive, HER2 enriched, and then we'll call it triple negative or basal-like, even though I just told you that's imprecise use of the word basal. And you can skip over all this, and I send you to the reference if you're interested in this. But this is the number of mutations per megabase DNA mutational analysis. And what you see, I don't have my glasses on, but basically that the number of mutations is 0.84 per 100 MBs in the hormone sensitive. It's a little higher in the luminal B. It's 2 mutations per MB for the HER2 enriched, but triple negative, and again, it's just basically dependent on the size of the bar here. The longer the bars, the more mutations. Basically, you can see that triple negative has a lot of mutations. These aren't just BRCA mutated triple negative. This is just a general subset of triple negative. So we know that triple negative has a fair amount of mutations, and the premise behind the trial is that triple negative has a high mutational frequency, in part because of homologous recombination defects. That means these tumors can't repair their own DNA. And this was data from the BRCA space of PARP inhibitors. And what you can see here, and this was published in Lancet a couple years ago, is that these PARP inhibitors, this is response rate. And you can see that the objective response to the PARP inhibitor, which is one of the two agents in our clinical trial, was about 41%. This is pretty impressive considering that these are, for the majority, are triple negative patients, and I just told you the highest response rate we've ever seen is 25%. So this is a subgroup of triple negative in BRCA mutation carriers. These drugs, which are oral agents, appear to have a fair amount of activity. So that's just a fact you have to kind of stick in your head. But the whole premise behind the trial is that there is non-BRCA mutation triple negative breast cancer that also cannot repair its own DNA. And many people are looking at this area. And at least in my practice, because olaparib is commercially available for BRCA mutation carriers, we see a high response rate in these very, very aggressive triple negative breast cancers. There must be a timer on my slide set, sorry. That basically makes them exquisitely, so these tumors are sensitive to PARP inhibitors, but they're also sensitive to platinum chemotherapy, in particular cisplatin or carboplatin. And so in non-BRCA, what we refer to as sporadic triple negative breast cancer, we believe that there is a homologous recombination defect that we don't know what gene or what mutation is driving it. And so the premise of the trial is that we're going to take patients, triple negative breast cancer patients that have a response to platinum. It's like the poor man's test of whether or not these tumors can repair their own DNA, and then treat them with PARP inhibitors to see. So we're taking a subgroup of triple negative, and instead of, we're going to do an outback molecular analysis, but we're treating with platinum almost to see if they cannot repair their own DNA because they would be sensitive to the carboplatin. I'm going to show you the design in a second. And then the other component of this trial is an immune therapy, and for the sake of time, I'm done. Sorry, I have no idea what's happening with my slides. I have a feeling there's a timer. But so the second part of it is immune therapy, and let me just get to the trial design for the sake of finishing this talk. So here, I hope it stays on the screen, is the DORA study design. This is going to be a 60-patient study, global study, based here in the DCRI, of metastatic triple negative breast cancer. We're going to take patients who have received platinum and did not progress within the first 12 weeks. And there is a population of triple negative that progresses, and we think it's about 1 out of 4 of these patients will never demonstrate sensitivity to platinum. And then we will randomize them to either a PARP inhibitor till progressive disease or the PARP inhibitor or olaparib with immune therapy. And for the sake of time, I couldn't get into a lot of the details. But if the PARP inhibitor inhibits DNA repair, your mutational frequency is going to go up. And therefore, immune therapy should work better because you have a higher antigen load for the tumor to express, and therefore the immune system to recognize and hopefully augment. And so that's the premise behind the two arms of the study. So triple negative with an agent that is very active in BRCA-driven cancers. We're selecting tumors that have BRCA-NUS, even though there's not a known BRCA mutation, treating one group of these women with a PARP inhibitor and the other group with the PARP inhibitor plus immunotherapy. And this was, to me, is the kind of trial that would be easily augmented by a supercharged investigator-initiated study. The company will get some benefit out of it. They'll get data. They'll get thought leaders. I don't know what else they'll get out of it. But the patients will get a novel trial that probably wouldn't be a first-pass industry study. And then the investigators really think this is an important question to ask, both in terms of immunotherapy, but also PARP inhibitor in non-BRCA mutated cancers. Sorry about that. And probably the usual suspects as far as endpoints. And the other thing that this collaboration has allowed is that Rebecca and I were able to apply for a collaborative grant from NUS and Duke through the Global Health Institute. So we were awarded another grant outside of the clinical trial grant to be able to do some of the science. So this is just the schema for the tissue that's being collected. Slides are being collected, germline DNA is being collected, serum's being collected for, I didn't even have time to talk about the cell-free DNA mutational analysis that will try to match up with the tumors in this particular population of patients. Very exciting types of work, and we have a different schema for who's doing what. A lot of the homologous recombination defect is going to be done in Singapore. A lot of the mutational exon sequencing is going to be done here at the Duke Cancer Institute. And this was just a very first pass, I think, demonstration. If we could figure out how to make these types of collaborations work in cancer, there is a lot of support for them both here and in Singapore. This is what NUS would like to see the relationship with the Duke Durham kind of evolve into. Here are the sites, so Duke and Memorial Sloan Kettering, Singapore, and then 2 sites in Korea that Rebecca and I have collaborated with extensively in other clinical trials. This is the timeline, and we'll skip over that. And here are the conclusions, just because I don't want anyone to look at it that closely. But hopefully what I've shown you, and I went a little longer, and I'm sorry about that, but you can see I'm pretty excited about this collaboration, is that new therapies are needed in triple-negative breast cancer. So the need is there. When anyone ever comes to me and wants to discuss their new drug company or their new trial, I always say, tell me what the need is. Because right now in breast cancer, we're going to have 4 CDK inhibitors that Pfizer, Novartis, and Lilly spent billions of dollars developing, and they're all for the same patient population. So I'm not so certain that we needed to do that, but that's how it happened. That novel clinical trials are necessary. I think we have to change the whole way that we do trials in cancer care in particular. And that the collaboration, hopefully the DORA study will be the first pass in figuring out a way to do these type of supercharged investigator-initiated studies that I think have worked very well within the DCRI and other disease types, and establish a correlative science platform. And although this has been a lot of work, it's been very few people that have been instrumental in getting this project off the ground. Obviously, my collaborators in NUS, Rebecca and Tira, who's one of her fellows that has done a lot of the work on this project. And we've had a lot of fun doing it. Trish, who's the project manager, who was very brave and either volunteered or is being punished to be the project manager on this project, has jumped right in with 2 feet, because this is kind of a wacky, unproven territory, and we're all going to have to be fairly flexible in figuring out solutions. I'm saying that to myself as well. And Andrew and Martin, who, when I first approached them with this idea, were very supportive the entire way and listened to my crazy ideas and put up with some really crazy questions. And then I think part of the success is that we've had an advocate within the pharma who sat down and listened to us. And so every time this was presented at every committee, and there was a whole nother team at AZ, imagine we had the immunotherapy team and the PARP inhibitor team. These were 2 separate, we might as well have been dealing with 2 different companies. That's how far apart they were. Really identifying someone who was willing to advocate for us within AstraZeneca, who is the sponsor of this study, and hear in particular about our ideas of why we needed so much money to do an investigator-initiated study, but also that we're hoping that they will view this as a success and want to do other trials in this space. And hopefully this trial will serve as a model for other drug companies who find that this particular type of effort is worth investing in. So I'll finish with that, and we have just like a few seconds for questions.
Trish, are we still using that investigational product in the study?
Yes.
Okay.
Durva.
Because that wasn't up
Here
there.
with the question was.
Durva. So this is David Jensen, who's going to be submitting your IND.
Oh, yay.
No, I was asking because in your slides you didn't highlight a third compound that's in the study.
Oh, the third. No, it's 2.
Just 2, but
It's
they're
a lacquer
not an
Durva.
approved agent.
Right. Okay. Just clarifying.
One question. So you talked about the challenges of conducting this work at a cost that is fundable and reasonable. Have there been efforts to conduct pragmatic clinical trials in this space, or is that a strategy that wouldn't apply complexity of data that's needed and perspective.
Help me understand what you mean by pragmatic trials.
We're doing a lot of work that takes advantage of data that's collected as part of routine healthcare, and also using that as a recruitment environment from a sort of national perspective, particularly for rare diseases. So in the cardiovascular space, it's promising because it means that not every trial is sort of reinventing the wheel, and
right.
There isn't so much redundancy in the conduct of those projects. So it seems like there could be promise for that type of approach here.
Yeah, and I think that's the idea behind big data in the cancer space. And obviously one of the 6 priorities for the moonshot is to collect clinical data to look at outcomes research. I don't particularly know how you do that when you're putting a new drug into a new disease. I mean, you can think of this as a new disease. I couldn't get either one of these drugs for my triple negative patients right now. So, and the immunotherapy space, even though it's so promising, and I think it's okay to say this, the companies are developing them in spaces where there's no approved comparator. So they're getting approval of single-arm studies, bladder, thyroid, where there's a recognition that there's not an acceptable standard. If you came to me with $1 million tomorrow and asked me to treat you with immunotherapy, Duke would not even allow me to do it. Now, they would probably take the money, I don't know, but they wouldn't allow me to administer it. So I think moving forward, we'll have that. I think that the companies are going to be forced to share some of this cancer data. I mean, I think that's going to be one of the really positive, when you think of all of the big data companies in the cancer space. But I think we have a long way to go. It's not cardiovascular disease. So if you ask me, and one of my roles within the DCI is strategic planning, and if you ask me how many molecular profiles of any sort have been sent on the tissues that were collected here at Duke, i.e. patients that had tissue collected here, how many of them have had any form of genomic sequencing at all? I couldn't answer that question even with EPIC. So right now we're hand extracting data based on send out lab results. I mean, that's really, I'm just being very practical. So one of the things we're trying to do, like they did in Singapore, is do a molecular tumor board so that across the board everyone gets molecular profiling and you have that data, and then it could be shared in the context of the company that's studying the drug in that patient population. We're not very close to that. I hate to be so depressing. Even the institutions I think are the furthest along, which would probably be Memorial and MD Anderson, we're off of pure philanthropic support, like I had $150 million from Revlon, let's say. They are offering molecular profiling, but even then, only 5% of their patients go on trials. So 14,000 patients at Memorial each year, 10,000 are getting, I'm just going to make that number up, 10,000. So 5% of 10,000, and then they're going on a variety of studies because, like it or not, we still do lung cancer trials only in lung cancer patients. And so I think I think the way we're going to come up with solutions is if we think of cancer as a very unique, the solutions that have been found in cardiovascular are not going to apply to cancer. I hope I'm not upsetting anyone. But it's just a whole nother layer of complexity. You have to gather tissue, and the outcomes aren't, did you have another heart attack or not, or did I don't know, I'm gonna make it up, your viral load come up to a certain level within a period of time, and that's because of the heterogeneity of cancer. And that's where big data is gonna help us if we can get our act together. Thank you guys very much.
Thank you.